and somebody tell me if it doesn't work. So hopefully you can maybe give me a thumbs up, Ridwan, just to say that you can see me. Good.
Yeah.
Okay,
Yes,
that's good. Okay. So thanks for asking me to speak today. I'm going to be talking about diagnosing the descent of Parkinson's with mass spectrometry and joy. And this is a rather crazy project that I became involved with. And if we just look at this picture, just to kind of how things started, this is a picture of Joy Milne. Joy is a retired nurse from Edinburgh, and Joy is sitting in front of a mass spectrometry machine, and she's smelling the chemicals as they come through, and she's pressing a button when she thinks she can smell Parkinson's disease. So this is a rather crazy situation. And what was it that Joy was smelling, and how did we get here? And so to understand kind of how this whole thing started, we have to go back quite a while. We have to go back to 1982. And this is a picture of Joy in the center of the picture, and her family with her husband Les on the left side of the picture. And Joy is what we call a super smeller. These people kind of exist in the population, and they have better senses of smell than the rest of us do. And often they work in, the wine industry, the perfume industry, and so on. And it was around this time in 1982 that Joy noticed an unusual musky smell on her husband Les. Not an unpleasant smell, but what she describes as a musky smell. So she kind of said, have you changed your aftershave? Are you using different shower gel? And Les goes, no, I'm not. And eventually he gets a bit irritated by these questions. And so Joy stops asking them. But this musky smell continues. And then we move forward 12 years. So that's a key point here, that he had this musky smell for 12 years before anything else happened. And then he starts to develop some neurological symptoms, and he gets diagnosed as having Parkinson's disease. And like many people with Parkinson's, he does pretty well for a number of years. And we move forward to 2009. And Les is now a little more disabled by his condition. And Joy and Les are members of Parkinson's UK, which is the main UK Parkinson's charity. And Joy's noticed, when she's gone to clinics with Les, or when she's gone to Parkinson's UK meetings, she's noticed the same musky smell on other people with Parkinson's disease. And she wonders, could this be a symptom of Parkinson's? And it was in 2012 that at a Parkinson's UK meeting, that this chap here, Tilu Kunath, is giving a talk. And he's actually a researcher from Edinburgh, and he was giving a talk on stem cells in Parkinson's disease. But at the end of the talk, Joy put up her hand and she asked him whether people with Parkinson's have an unusual musky smell. And Tilo had never heard of this before, but to his credit, he decided to take Joy seriously, and he was intrigued that she thought that she could smell Parkinson's disease. And so what he did was he set up a small study, and he got 12 people to wear T-shirts overnight. 6 people with Parkinson's and 6 control participants without Parkinson's, and they wore T-shirts overnight. And in the morning, he gave all the T-shirts to Joy, and he said, okay, if you think that you can smell Parkinson's disease, tell me which T-shirts were worn by people with Parkinson's disease and which T-shirts were worn by the controls. So Joy got all 6 of the Parkinson's T-shirts correct, and she got 5 out of 6 of the control T-shirts correct. So she did incredibly well. She got 11 out of 12 T-shirts correct, and therefore we thought this is clearly something in this. Of course, she got 1 wrong, but still 11 out of 12 is pretty good. But what about this one that she got wrong? This was a guy called Ken, and he was one of the control participants in the study. But Joy thought that he had Parkinson's disease. But interestingly, about 9 months later, he developed some mobility symptoms, and he was in fact diagnosed with Parkinson's disease, and he contacted us to tell us that this had happened. So Joy, amazingly, was able to diagnose him with Parkinson's even before he developed any mobility problems. And so this told us really there must be something in this. So there must be some change in the skin chemicals which is happening maybe quite a long time before people develop Parkinson's disease. And here is a picture that Joy herself drew. And this is a picture of basically how smells are to her. So she walks into a room and it's like she can see the smells. It's almost a kind of synesthesia. And you can see different smells coming from different parts of the room, and a little bit like what we're seeing in the picture. This is kind of how a room is to her in terms of all the different smells coming from different parts of the room. And when we think about smells, of course, we think about sweaty armpits and so on, but actually that wasn't where Joy felt the smell was coming from. She felt it was coming from the neck and it was coming from the upper back. And we think that the smell was coming from sebum. Sebum is this oily stuff that we have on our skin, of our foreheads and our upper backs. It causes acne and so on. And it's been known for almost 100 years that sebum is altered in Parkinson's disease. You get seborrhea, abnormalities of the skin, greasy skin, increased and altered sebum production in Parkinson's disease. And we think that it's within the sebum that the chemicals are changing, and that's what Joy is smelling. And so around this time, Thilo Kunath in Edinburgh thought, this is interesting. Maybe there is maybe there is changes in the chemicals of the skin in Parkinson's, and maybe we can use this as a diagnostic test. And so he contacted Professor Perdita Barron, who's a professor of chemistry here in Manchester, and one of the world experts in a type of chemistry that we call metabolomics, which I'll talk about in a minute. And Purdy contacted myself as a kind of Parkinson's researcher, and we got together, myself and Perdita and Thilo and of course Joy, and we discussed whether we might want to set up a research study. A few questions that we wanted to answer. Firstly, are the skin chemicals different in Parkinson's disease? And clearly this seems likely. Joy has noticed an altered smell, and so it suggests that the skin chemicals are different, but can we prove this? And also, can we use this to develop a simple, cheap, cost-effective kind of diagnostic test, just a simple skin swab as a way to diagnose Parkinson's disease? And finally, can we use this test to diagnose the condition much earlier than is currently possible? Remember, Joy can smell these alterations 12 years, maybe longer, before any mobility symptoms arise. So maybe we can use this test to diagnose Parkinson's much earlier than is currently possible. And so we applied for funding to Parkinson's UK and the Michael J. Fox Foundation, and we were lucky enough to get funding from both of these organizations. And this enabled us to set up a large research study to investigate all this. And the study ran initially at 25 centres around the UK, and some of the data I'm going to present, at least to start with, is from the first 64 participants. But we now have swabs taken from over 1,000 participants in this study, and we've moved into other countries, including some countries in Europe, and some countries in North America, and some countries in Africa. And what we did in the swab collection, just take a simple gauze swab, or more laterally using just a simple Q-tip, and swab in the areas that you can see on the picture there. Mainly, we can swab on the forehead or on the upper back, although the upper back is a bit of easier place to use because people wear makeup and so on the forehead, and that can complicate things. And so most of the swabs from the upper back. And the swabs are simply posted at room temperature, first class cold post to Manchester. So they don't have to be kind of frozen en route, which is obviously useful for moving into more rural settings and so on. And then when they get to Manchester, it's that at that point that we would freeze them down until ready to use them. And then the samples go through a process called metabolomics. Metabolomics is a scientific way to look at the chemicals in the swab, look at thousands or hundreds of thousands of chemicals, and look at whether there's differences, for example, between Parkinson's and the control swabs. Metabolomics you can use on skin swabs, you can use on blood, you can use on urine, and so on. But the type of metabolomics we were using, of course, was on the skin swabs. And this is basically fancy chemistry to identify all of the chemicals in the swabs, and then fancy statistics to identify which chemicals are different in Parkinson's versus control. One of the nice things really about doing research like this and collaborating with different people in different disciplines is I've got to learn quite a lot about chemistry and statistics and metabolomics, which has been interesting, different from my usual kind of neurology, which I do in my day-to-day life, and also to get taught by really some of the world experts in these areas. And so what I'm going to do for the next few slides is just tell you a little bit about metabolomics and how the chemistry and the statistics and so on work. If you're not particularly interested in that, then you can go to sleep for a few minutes and then wake up when I come to present the results again. So, as I said, metabolomics involves using fancy chemistry to identify the volatile chemicals in the swab. In the initial experiments, we were looking for volatile chemicals. That's chemicals which can get into the air and become a gas, if you like, because we knew there had to be some changes in the volatile chemicals on the skin swabs because Joy is able to smell. And if she can smell something, it has to be able to get into the air. And we used this technique called thermal desorption gas chromatography mass spectrometry. So what on earth is that? And I think the easiest way to explain how this works is to think of it as a kind of 100-meter race. Imagine, if you like, that there's a 100-meter race going on outside the window. The curtains are drawn, so you know the race is going on, but you can't see it. And you can hear the gun go bang when the race starts, and you can hear another gun go bang when somebody crosses the line. And your job is to say who it was that crossed the line just based on how long it took them to get there. And there are three people in this race. There's this chap here, Usain Bolt, the 100-meter world record holder. There's this chap here who looks with his Zimmer like he's not going to be particularly fast. And there's this chap here whose athletic days are sadly quite some time behind him. And so bang, the gun goes. And then 9.82 seconds later, bang, the gun goes again. And you think, I know who that was. That was Usain Bolt crossing the line. Then a few seconds later, maybe 13, 14 seconds, I'm not really sure, bang, the gun goes again. And you think that was probably me crossing the line. And then after maybe 23 seconds, bang, the gun goes again. And that was probably the old chap with his Zimmer kind of hobbling across the line. So the idea here is that even though you didn't see anything, just by knowing how long it took the person to finish the race, you know exactly which person it was. And that's basically the idea of thermal desorption gas chromatography mass spectrometry. And I'll show in this slide a little bit more about how that works. So on the left side of the screen, we have all the chemicals and they're sitting in a test tube. The skin swab, basically a little bit of the swab is cut out and put into the test tube. And we heat the test tube and all the chemicals kind of, because they're volatile chemicals that we're looking for, all the volatile chemicals come off the swab and they stick to this thermal desorption area, just a place where the chemicals stick to. And this is basically the start line of our race. Then what we do is we slowly heat up this area so the different chemicals come off it depending on how volatile they are. And so it's almost like some chemicals are getting a head start. Some chemicals come off a bit quicker than other chemicals. And then they run through this gas chromatography column, and they run through it at different rates, depending basically on how hydrophilic or hydrophobic they are, how much they love water, how much they hate water. They come through at different rates. So all the chemicals are getting separated in time. Some chemicals arrive some minutes after other chemicals. They then go through the mass spectrometry machine, which I'm going to talk about on the next slide. And then they arrive at their detector. And different chemicals come at different times. So you can see what you get is a reading like that with the time in minutes on the x-axis, and then how much of a chemical is coming through on the y-axis. And so you know, you can actually know fairly accurately which chemical you're dealing with, just depending on how long it takes to get through that system. And clinicians, if you work in hospitals and you've sent off, for example, a urine toxicology screen looking for drugs, or a plasma organic and amino acid screen, this is exactly the same type of process that they use to tell you what chemicals are there. So now we know a lot about the chemicals, but there's still this mass spectrometry machine that we need to talk about. That tells us with a high degree of accuracy which chemicals we're actually dealing with. And what a mass spectrometry machine is basically an incredibly accurate weighing machine. So imagine, if you like, that I get everybody in this talk to stand on some scales and I take down a reading about how much you weigh. And then I kind of go out the room and I say, okay, I want one person to stand on the scales and I'm going to tell you who that was. I can look at my table of all your weights and I'm going to tell you who it is. So somebody stands on the scales, I can't see them, and I look at my table and it says they weigh 70 kilograms. Well, that could be probably quite a few of you. I don't really know who that is. But if it says that they weigh 70.12649839274 kilograms, well, I know exactly who it is. If you get a weight at that level of accuracy, I can say exactly who that person was. And that basically is the idea of mass spectrometry. If you weigh a chemical incredibly accurately, then you can tell exactly what chemical you're dealing with. And here's how it works in kind of simple terms. So remember, the chemicals have all been separated in the gas chromatography, so they're coming through at different times. And then we enter the mass spectrometry machine, and on the left side they enter. And what first of all happens is they get bombarded with electrons. So remember, different chemicals are coming through at different times. They get bombarded with electrons, and that acts like kind of billiard balls to knock all the electrons out of the chemical. It's like a hailstorm of electrons, which knocks all the chemicals out, or knocks an electron out of each chemical. So the chemicals become positively charged. And then they get fired through an electric field, and they get deflected by the electric field because they are positively charged. And if you think about deflecting something, for example, imagine you're playing baseball and someone throws a cannonball at you and you whack, you try and hit it, you're not going to be able to deflect it very far. But if they throw a tennis ball at you, then wham, you can score a home run. And so the distance that something gets deflected is related to its mass, or technically its mass charge ratio. And so the first chemical comes through like so. It's quite a big chemical and doesn't get deflected very far, and it ends up there on the detector. The next chemical comes through, it's a slightly smaller chemical. It gets detected a bit further and it lands there on the detector. And the third smallest chemical gets deflected a bit further and lands further down. And so basically, we have weighed the chemicals by demonstrating how far they get deflected. And then we can really determine exactly what we are dealing with. It's worth pointing out that modern mass spectrometry machines don't quite work like this. This is the kind of slightly old-fashioned machines. Modern, what we call quadrupole mass spectrometry machines work slightly differently than this, but basically the principle is the same. We deflect chemicals, and the size of the chemical is related to how far they get deflected, and so we basically get a very accurate mass. And that's the whole process, basically. The chemicals are separated in time using the gas chromatography, and then each individual peak we take and we run it through the mass spectrometry machine, and that gives us a very accurate weight, or more accurately, a very accurate mass charge ratio. And that basically tells us exactly what chemical we're dealing with. And we do that over hundreds and hundreds of chemicals, or even thousands of chemicals. And then we can see which chemicals are different in the Parkinson swabs versus the control swabs. And then the second thing that we do is fancy statistics. And I'm not going to spend very long on this, but just to tell you a little bit about the sort of statistics that we would use in order to, if anyone reads any of our papers, for example, just a little bit of background knowledge and what some of these statistics mean may be a little useful for you. And one of the things that we use is a thing called permutation testing. So the idea is this: if you look at hundreds or thousands of chemicals in the skin swabs from the Parkinsons and the skin swabs from the controls, you're looking at so many chemicals that maybe just by chance, you might pick up a difference between the two groups when there's no real difference. So what permutation testing is doing is to say, is there a real difference between these two groups? Are they definitely different? And the way we do that is we look at the skin swabs, comparing Parkinson's and controls. You run all the tests. We look and see how accurately can we separate these two conditions based on the chemicals in the skin. And then we basically jumble all the samples up. We get a whole bunch of controls and a whole bunch of Parkinson's, and we call that nonsense group 1, and a whole bunch of the other half of controls and the other half of Parkinson's, and we call that nonsense group 2. And then we see, can we separate these two nonsense groups? So using the chemicals in the skin, are we able to separate these two? We've got hundreds, even thousands of chemicals. Can we separate these two groups? Can we see differences between these two groups? Then we look at nonsense group 3 and nonsense group 4, just generally splitting up the groups into kind of random groups, mixtures of Parkinson's controls, and seeing if we can separate and see differences between these two kind of random groups. And what we want to see is that we can show clear differences between the Parkinson's group and the control group based on all the hundreds of chemicals that we're looking at, much more accurately than we can when we use the different nonsense groups. And that's what we call permutation testing. And that tells us that we've got a real difference between the groups. It's not simply that we have such powerful tests and statistical techniques that we get some random difference. And so now on to the results. When we do all this, when we do the gas chromatography and the mass spectrometry and all the fancy statistics, we find that yes, we can distinguish the swabs, the skin swabs from Parkinson's compared to the skin swabs from people without Parkinson's. And we can actually do this with 90% accuracy. So really pretty good that we can diagnose with 90% accuracy whether somebody has Parkinson's or not, simply based on the chemicals in their skin swab. Obviously, it's not 100%, and you probably need, to look at other things like clinical symptoms and maybe one or two other pieces of information in order to get up towards 100%. But really, for something which is very simple, just a skin swab, there's no needles, the storage of the samples is pretty easy because they can be sent at room temperature. It's amazing that we can get such an accurate result. And these are the structures of 4 of the chemicals which are different between Parkinson's and controls. They're mainly lipid chemicals. They've got big long names like pyrylic aldehyde and eocaine and hippuric acid. The structures are there if you are a chemist and you're interested in that type of thing. But basically, these are a lipid type of chemical that we're finding on the skin, which is different between the skin swabs of Parkinson's and the skin swabs of the controls. And now the picture that I showed you at the start of the talk, we were able to turn the thing round a full circle and get Joy to come back into the lab and see if she could validate what we had done with the chemistry. And what we did was we got the mass spectrometer and the gas chromatography machine. So the chemicals are coming through the gas chromatography machine. They're coming through at different times, remember, they've been separated in time. And we have a kind of Y port. So some of the chemicals get diverted to this kind of nose port there where Joy can smell them. And the other half of the chemicals get diverted to the mass spec machine for further analysis. So Joy kind of smells the chemicals as they come through and presses a button when she thinks, that's the smell of Parkinson's disease. And amazingly, Joy presses the button at exactly the same time as the chemicals that we had identified were coming through. And so we're getting, in a sense, a second validation from Joy's nose that what we are picking up in the chemistry is the same as what Joy is picking up with her nose. So we published our first paper a couple of years ago now in this journal, ACS Central Science, which is a high impact chemistry journal. And the journal contacted us maybe 2 or 3 weeks before the paper was going to be published to tell us that they wanted to run a press release. They thought there might be some media interest in our study. And I didn't think much of it at the time. I just said, okay, fine, whatever. If you want to run a press release, that's great. So then the paper comes out and the press release gets run and all hell broke loose. All of a sudden, I found myself at the center of a media storm. There was just a ridiculous amount of media interest in our study. A couple of weeks after the paper was published, the journal contacted us to tell us about this. This is a thing that we call the Altmetrics attention score. It's a kind of score that the journals use to kind of quantify the media interest in their papers. It looks at, for example, the normal media, the old-fashioned media like newspapers, television, radio, and so on. And it looks at the modern media like Facebook, Twitter, Instagram, Reddit, and so on. And it combines everything into a score regarding how much interest there has been in a paper. And our paper scored 1133, which was the highest score this journal ever had. And this is a high impact chemistry journal. There'd never been more than 1,000 before. And so there really was enormous media interest at the time. And for me, this all kind of culminated with an appearance on the BBC Breakfast Sofa. Some of you may know about this. This is the main UK breakfast television programme. It's got an audience of several million people. They have this iconic red sofa, which you can see me sitting on. And you can see me on the left beside Joy Milne, of course, the super smeller, and the two presenters from the programme. And I must say that sitting on live television in front of several million people is easily one of the scariest things I've ever done in my life. And I think it's nice to do once, but I don't plan on doing that again. So what next for us? I've obviously talked about studies that are being done and be published a couple of years ago. Things did slow down a bit. Obviously, during COVID, the whole lab closed down and we probably lost a year to 18 months of the work. But we are kind of back up and running now, and we've got actually more than 2,000 participants in the study now. And we are starting to move into other countries, including North America, Europe, and Africa, that we're collecting samples from. And one of the things that we want to do, as well as developing an improved and a cheap and a simple diagnostic test in Parkinson's, one of the things that we really want to do is see if we can look at prodromal Parkinson's. I'm sure most of you know, or many of you know, that Parkinson's is thought to start some 12, 15, if not more years before people present to their doctor with mobility symptoms. So only when you get to what we call stage 3, 4 disease, that the substantia nigra starts to degenerate and people start to develop kind of mobility problems like tremor, stiffness and slowness and so on. And we talk about a prodromal phase of Parkinson's where the neurodegeneration has started, but clinical Parkinson's is not apparent yet. And if we look at, for example, the dopamine neurons in the substantia nigra, we can see they're gradually reducing over time. And clinical symptoms are gradually coming on over many, many years. And when we currently diagnose Parkinson's disease, when someone presents to their doctor for the first time with mobility problems like tremor, stiffness, slowness, and so on, roughly 60 to 70% of the substantia nigra neurons have already degenerated, even when they first present. And what we really want to be able to do is use a skin swab test to try and enable a much earlier diagnosis, a prodromal diagnosis in Parkinson's disease. Because remember, Joy can smell this change in chemicals some 12 years before her husband Les develops Parkinson's disease. And so we have every expectation that the skin swab can be used a lot earlier than simply after the diagnosis has been made. And we're hopeful that if we can make the diagnosis a lot earlier, this is going to enable improved neuroprotective treatments in Parkinson's disease. There's a bunch of treatments coming into clinical trial at the moment, which we hope will be neuroprotective, which may slow down or halt or even reverse the progression of neurodegeneration in Parkinson's disease. But they're much more likely to help if you can pick the disease up at an earlier stage. If we look at treating cancer, for example, if cancer is spread 70% around the body, it's pretty difficult to treat. But if you can pick it up at a much earlier stage, then of course cure becomes possible. And we hope, therefore, this simple skin swab based on the simple observation from Joy can actually help us to move towards earlier diagnosis and better treatment in Parkinson's. And another direction that we've moved in involves this paper that we published last year in Nature Communications. And what we did in this paper is slightly different. We talked about how we looked in the initial study at volatile chemicals, chemicals that get into the air, chemicals that you can smell. And we used this fancy technique of this thermal desorption, gas chromatography, where all the chemicals were heated and they came into the system that way. In this paper, we used liquid chromatography. So basically, you just take the skin swab and you put it in a solvent, and you can basically get all the chemicals out. And so we can get much higher numbers of chemicals in the swabs when we use liquid chromatography. And we run it again through this machine, and we look at thousands of chemicals really, and we see what differences can we see between Parkinson's and control. But now we're looking at much larger numbers of chemicals. And what we then did was use basically chemistry software. We could take all the chemicals that are different between the Parkinson swabs and the control skin swabs and run it through this kind of chemical database software. And it will tell us which metabolic pathways are altered between the two groups. And what we see is that certain metabolic pathways, including the carnitine shuttle and certain mitochondrial pathways, are dysregulated in the skin swabs when you compare the chemicals in the Parkinson's group from the chemicals in the control group. And it's amazing because when other people have, not ourselves, but other people have done kind of biochemical studies of postmortem brains in Parkinson's disease, they see very, very similar chemical changes in the brain that we are seeing in the sebum on the skin. So it's almost like by swabbing sebum, we're getting a window into the dysregulation of mitochondrial metabolism in Parkinson's disease. When I first saw this data, I was a little bit skeptical. I thought, hold on a minute, there's this thing called the blood-brain barrier. How are you going to get chemicals that are abnormal in the brain getting through the blood-brain barrier and then pick them up on the skin? I don't really know how that could happen. But of course, these are lipid chemicals, and lipids don't really care about the blood-brain barrier. Lipids can just move down their concentration gradient from areas of high concentration to areas of low concentration. And so if you get altered lipid concentrations in the brain, it's very realistic to expect that this could appear as altered lipid concentrations on the skin. So we haven't really touched the surface of what we can do with this. Now we can actually, in a sense, analyze brain biochemistry simply by swabbing the sebum on the skin. And this leads me to just the final thing to think about is what about the whole idea of using sebum as a diagnostic biofluid, this greasy stuff that we have on our skin. There's this idea that chemicals kind of get trapped in there, a bit like a bird being trapped in a cage. And so, for example, if you're checking for diabetes, when you do a random blood glucose, you might miss it. Even if someone's got diabetes, it might just be normal at that particular moment because it's going up and it's going down all the time. But if you check an HbA1c, you get a kind of much longer term marker of what the glucose has been doing in the blood over the last several weeks. And that's a much better way to diagnose the condition than a random blood glucose. And maybe sebum can be the same sort of thing. We could imagine chemicals going up and down in the blood all the time when we do a quick blood test and we miss it. But the chemicals kind of appear in the sebum, they kind of get trapped there and they stay there for weeks and weeks and weeks. And maybe therefore we can use sebum as a whole new way of diagnosing not just Parkinson's disease, but other conditions as well. The lab that I've been collaborating with, they had to develop a whole kind of whole process basically to use skin swabs and get them into a mass spectrometry machine. You could imagine the greasy stuff is kind of blocking up the tubes of the machine and so on. And so they had to develop a whole kind of protocol really for how to do this. But now that they have the protocol, maybe we can use sebum as a new novel diagnostic biofluid. Interesting that the lab's been working on this over the last couple of years, and we've shown that we can diagnose tuberculosis just by looking at the chemicals in the sebum. And interestingly, we can diagnose COVID just by looking at the chemicals in the sebum. And so this may be a whole, just by accident really, through serendipity, we might have stumbled across a whole new way and a simple way of diagnosing disease. And that's all I'm going to say today. I just want to thank Professor Barron, the Professor of Chemistry that I work for, who's basically been the chemistry lead for this whole project. Also Ellie Sinclair, the postdoc, what we call the wet chemist in the lab. That means Ellie's the person that did most of the mass spectrometry and chromatography analysis with the chemicals and the swabs and so on. And Drupad Trivedi, the postdoc, who's the dry chemist in the lab. So he's done most of the statistics and so on with all of these analysis. And of course, I want to thank the principal investigators at all the sites around the UK and also around Europe and abroad, and all the patients who kindly agreed to be participants in our study and help us by donating their skin swab samples. Thank you.
Thank you so much, Professor Silverdale, for the wonderful talk. So there are a lot of questions based on from Facebook and on Zoom. So let me take the Facebook. Oh, let me take the Zoom one first, then I go to Facebook and alternate in between. Someone says, have you been able to identify other persons who are able to smell the Parkinson's? And we know that quadriplegics also have a very specific aroma or smell on them.
So yeah, in terms of the first bit, yeah, we a lot of people have contacted us and said that they think they can smell Parkinson's as well. We haven't sort of tested them. We had this idea of maybe testing them, maybe sending them out some samples and saying, you know, okay, there's 10 samples, which people is Parkinson's, which isn't, and seeing if they got it right. And then the people that were right, we thought about maybe inviting them to the lab. And we have this idea maybe to do that. And at one point, the TV was interested in maybe filming some of this, but we haven't gone through with that yet. So far, we haven't really used the human, other human smellers, but certainly lots of people have contacted us.
Yeah, there's someone raising their hand in the among the participants. Let me take the questions first, then I can go to him. So someone says, thank you very much for your talk. Any idea on interest in also knowing what could be special about Joy's nose? Perhaps they could influence future diagnostic approaches to these and other related diseases.
No, that's an aspect we haven't really looked at. What are super smellers? What's different in their nose? We haven't at all looked. It's an interesting question, but we haven't looked for that. No, we have. I mean, just as an aside, we have looked at things like dogs, and we do have a collaboration with medical detection dogs to see whether we can train dogs to smell Parkinson's, and that's looking that we can. But specifically, what's different about Joy's nose compared to my nose, I don't think we have an answer to that one.
Let me take this from Facebook. So, very interesting talk. Were there any differences in the chemicals detected in the skin swabs from different cohorts or from different regions or different countries?
We haven't analyzed the non-UK samples yet. Where they're coming in, but they've not been fully analyzed yet. So that will be interesting to see whether we can pick up, you know, differences. We didn't pick up differences across the UK between different cities and that sort of thing, or really between different, you know, ethnicities, and we didn't pick up any differences there. But you know, we think, you know, different diets and so on surely would maybe cause some differences, but we don't have that data yet.
Yeah, someone said, have similar studies on sebum be performed in the context of other neurodegenerative diseases like Alzheimer's, schizophrenia?
I mean, as far as I'm aware, the lab that I'm working with are the only lab that's ever looked at sebum in disease, basically. We have talked about looking at other neurodegenerative diseases. We have samples from atypical Parkinsonian disorders like PSP and MSA that we hope to run on the mass spec machine. There's been talk about motor neuron disease, ALS, Alzheimer's disease, but we haven't done much of that yet. And this is all quite new. You know, we only did the Parkinson's stuff like a couple of years ago, and then we had COVID, and so then everything went on hold for a bit, and we're just getting back going again. But these are all things we would like to look at. Yeah.
Let me post someone that's raising their hand in the Sunday B song.
Say again?
Yeah, there's someone raising their hand in the among the attendees. I wanted to ask if you could type his question, or if you want to go and ask you personally. Dr. Sunday. Okay. Yeah, there's a question that says, can you place an age range at which positive detection can take place?
I think not really. No, we most of our participants have been fairly matched. We tried to match the control ages with the Parkinson's ages, and so most of our participants are, you know, at the older end, you know, more than 50 on the whole. We haven't looked at young skin swabs just because the controls were age matched, and we have one or two younger Parkinson's participants, but not enough to make any sort of answer to that question out of. So we don't really know. Certainly, when you look at blood metabolomics, there's a whole literature on changes that take place with aging and how you can perhaps diagnose frailty based on the blood metabolomics and so on. But we haven't looked at that age like that on the skin yet.
Trying to see questions from Facebook. Okay, yeah, someone says, how far do you think the environmental factors and air pollution can influence the nature of the detected chemicals in the skin swabs?
It's difficult to know the answer to that. I mean, all I would say is that we have 25 units across the UK, and when we run all the fancy statistics and see whether we can tell the difference between different units based on the skin swabs, some of them coming from, central Manchester, central London, other than coming from like fairly rural areas like Cornwall and so on, we haven't been able to distinguish the different units based on the skin swabs. So all I can say is from that, it doesn't look like things like the level of pollution and stuff. If one assumes the level of pollution in central London and central Manchester is higher than in kind of rural Cornwall, then we're not picking that up on the swabs.
Thank you. So question here, I think it says, what are the major metabolites or chemicals linked with PD or PD with Lewy dementia? Like if you look at it from your data, do you have any major?
Well, they are. It's all lipid chemicals. They've all got big long names: eocaine, perylic aldehyde, hippuric acid, and then some chemicals involved in the mitochondrial pathway, mainly lipids and things that we're picking up. But partly that's because sebum, I think, is largely made of lipids, and so that's therefore what you're going to get. But there's no kind of headline chemical yet. I think it's more of an overall metabolic change that we're seeing rather than just one chemical. There is some evidence that altered lipids can affect things like alpha-synuclein deposition, and people will look at alpha-synuclein deposition in a prion-like manner as maybe being the main process that goes wrong and causes Parkinson's. And there is some evidence that altered lipids can alter the deposition of alpha-synuclein. So it may all link in that way, but I couldn't give you like one or two chemicals and say that's the culprit. We don't have that data yet.
Yeah, let me ask mine. So earlier we were discussing about you having some of the studies in Africa. So I'm not sure if you have any samples from Nigeria yet, or if you have plans to collaborate with any Nigerian to send some samples to you too.
Yeah, we do. Well, we have a grant. There's a collaborator of mine. I'm just looking at because I have it here. Just thought someone might ask about it. Collaborator of mine, Richard Walker, who works in the northeast of England, but he also has for many years been working in Africa, and I think we talked about this before, looking in rural Africa and trying to find people with Parkinson's that haven't been diagnosed, and he's got quite a big program working that way. And now he wants to add skin swabs to that and see whether that will help. So we're doing it in Tanzania, Kenya, Ghana, Nigeria, South Africa, Ethiopia, and Egypt, looking at this list here. So that includes Nigeria. Yeah, but obviously we could still potentially look at other collaborations to get samples from Africa at some point.
Yeah. Someone says, are your participants sporadic PD or PD of genetic origins?
We haven't checked. I mean, most of them will be sporadic just by statistical whatever. This genetics not that common, but we haven't. I mean, that's an interesting thing to do, to look at genetic types of Parkinson's, and that is something that we want to do, especially, for example, LUC2, for example, a gene which puts you at high risk of getting Parkinson's, but maybe we can pick people up before they get Parkinson's and see whether we can see it happening before it happens. And it's an easy way to find people in the early stages of Parkinson's before they present with mobility problems. So we do want to look at all that, but we haven't done yet. One thing to add is that the mass spectrometry, when you're looking for one chemical, it's okay. You can make it quite cheap. But when you're doing this sort of big work that we're doing now, looking at hundreds of chemicals, it is quite expensive. So it all needs kind of grant funding and stuff like that. We can't just do all these things without getting funded.
Yeah. Someone says, bearing in mind the difficulty in replicating Joyce's nose, is there a possibility of developing test scripts for easy diagnosis? That's the first question. The second one is, does food ingested not alter these chemicals in the serum?
And so in terms of simple tests, that is the plan. Purdy, the chemist, describes it to me a bit like, you go to the airport and they do this thing with the swab and they put it in the machine and they tell you, I think they're looking for explosives or something, and they can do it really, really quickly using, it's an immune-based machine that works. And hopefully in the end, we can develop a very simple bedside skin swab test like that, that's not going to involve, you know, using a mass spec machine and stuff. Well, once we have identified a group of chemicals we're looking for. So that is certainly the plan, but we're early days for that one. And as far, I think you asked about diet. I think we haven't taken information about diet. I organized the initial tests and I tried to make the protocol as simple as possible, but I don't know if people that have organized studies where you recruit participants, like every 30 seconds that it takes to take extra of your time, or certainly every minute, probably takes like 5% off the number of people you recruit. So yes, you can ask all these questions, but then you might only get like 20 participants. We got 2,000 participants because we kept it really, really simple. It's like 5 or 6 questions and then a skin swab, and then they leave, and it's all very simple. So we didn't, by the way we designed it, we didn't get fancy information on diet and things like that. We want to do that now, but we didn't get that information first time around because we wanted to keep it as simple as possible to get as many people recruited as possible.
Let me see if there's any question from Facebook. It's not there. So no question on Facebook. Let me see if there's more. Then sorry. Okay, there's a question I missed. What influences your decision to evaluate the metabolites of skin secretions?
I suppose the answer to that would be joy. You know, we
Yes.
knew that she could smell this. The t-shirt test was quite impressive. She was telling us basically this is the area of the skin that it's coming from. We did speak to dermatology consultant about what do you think this is, and he said immediately sebum. This is the areas that she's talking about. These are the areas that have sebum, so I'm sure this is sebum. And so then that's why we went for that.
Okay, yeah, okay. Let me take this one. So there's some people that have this disease is called seborrhea, in which they have this itches on their skin. Is there a link between that and Parkinson's disease, or there is no link from the patient so far?
Well, Parkinson's, you get seborrhea in Parkinson's, as it looks quite similar to the seborrhea you get without Parkinson's. And I suppose the question probably that we need to know is, if you have seborrhea and you don't have Parkinson's, what do your skin chemicals look like? We have been collecting sebum swabs by collaboration with some dermatologists from people with seborrhea who don't have Parkinson's, who have got this greasy skin, and they go to hospital and see the doctor about it. We've kind of struggled to get decent numbers there. I think not that many people come to hospital medicine with that one, but we haven't got the answer to that yet. But we certainly are looking at whether, if you have seborrhea but you don't have Parkinson's, whether your skin chemicals resemble Parkinson's or not. We're starting to look at that, but we don't have the answer yet.
There's this very interesting one. It says, could it be possible to model Joyce olfactory senses so as to have other people smell diseases? Like, I think it's just a way of saying, is it possible to identify more super smellers in the population to help in the disease diagnosis?
I mean, we're using dogs. They may be easier because, but it is quite expensive to train dogs, as it turns out, like 20,000, 30,000 pounds a dog. So that's quite a lot of money. It's not going to be it's not going to be so many. There's people developing like electronic noses and things like that. And there's even a company called Mouse Sensor, which is developing mice to do this type of thing. I think in the end, it's all going to come down to the chemistry. I think the joy is an interesting kind of side issue, but we can't send Joy around all the clinics in the world. And so in the end, it's the chemistry. Is Joy started us on this road, but really I think it's the chemistry rather than the super smellers, which are going to finish really.
Okay. Yeah, this question, I think the person came in late. Thank you very much for the presentation. It's been very insightful. Is there a particular part of the skin the swab is taken from? I think those answered.
Yeah, so we need the areas that have the sebum, the greasy stuff. So basically forehead like that or top of the back are the two main areas that we have been using. Probably the back's better because makeup and things on the forehead just complicates things, whereas people don't tend to put makeup on their back. So mainly the upper back, just the behind the neck and shoulders, just anywhere there's grease on your skin, basically. We ask people not to shower on the morning that they have their swab done, so that they have slept overnight and woken up and the skin's a little bit greasy, and that's when we do it.
Let me check Facebook. Okay, we have 5 more minutes. If any more people have questions, please let us put them in the chat section. Yeah, I'm just curious about the diagnosis now. So if you want to make a simple material that could be used to diagnose this disease based on smell, so I'm just trying to imagine how it's going to look like. We know how pregnancy kit test is. You know, you have to drop urine. So what's the idea on how the stains, how it will be used to diagnose diseases going to be like? I'm just trying to.
I think there's two ways you could go with it. One is, like I said, this thing at the airport, you know, where they put this, I don't exactly know how it works, but they get a swab and they put it on your skin. You probably had it done. I've had it done a few times. You go through airports and they swab something on your skin, and, you know, like 20, 30 seconds later, they seem to be able to tell you whether you've got any explosives on you. And that uses a kind of very random, very quick chemistry test. So that would be one way of doing it. And I think it involves antibody-based testing, although that's different from what we do in metabolomics. I think in the end that involves more of an antibody immuno type of thing. Or the other way to do it is we can run samples very quickly in a mass spec machine, and so you could run many samples at once, and so it wouldn't be impossible to have a situation where you had 1 or 2 mass spec units per country just running these samples, you know, very quickly. You know, a bit like the COVID PCR test, where you have only a small number of centers, but they run thousands of tests very quickly. So that would be the other way of doing it. We haven't kind of got a definite idea on which direction to do that.
Okay. So our time is almost up. What's up?
Yeah. So would you like to provide kind of closing remarks for our audience before we close today?
Yeah, can you go ahead and do that, please?
Professor Monty, do you have any closing remarks?
Oh, me?
Yeah.
No, really, no. I think I've said what I wanted to say. It's lovely to speak to a different audience for me. So that's been really nice. Thanks very much for inviting me.
Yeah, great. So with that, let me thank Professor Monty for this wonderful talk, and also thank our audience for joining us today and making this webinar interactive. And see you in our next webinar, and bye for now.
Okay, bye-bye. Thanks very much.
Bye.
Bye.
Thank you.